Welcome to Unbiased Science,
where we bring scientific method to the madness.
We're your hosts, Dr. Jess Steier
and Dr. Sarah Scheinman.
And this week, we are tackling a topic that people ask about constantly in all different contexts, but the big C, cancer. And we are joined by none other than the Cancer Daddy, Dr. Joe Zundell. You do go by Cancer Daddy, right? Am I making that up?
No, you're not. First,
Is
thanks
that
for having
real?
me. The nickname actually came from Max Lugavere, believe it or not, which, it kind of stuck. He was just joking around. He'd said, there's people like in the wellness space that are like wellness daddy. Luke Cook is considered wellness daddy. And he just started calling me Cancer Daddy, and he said it in a public platform. And, when you have 1 million followers, then suddenly everyone just starts calling you Cancer Daddy, and it was really funny. So
Is that your Twitter handle?
No, it's first of all, I'm horrible with Twitter or X or whatever anyways, but it's it probably should be Cancer Daddy.
Well, anyway, so let me read this very impressive bio for Dr. Joe Zundell. So, and can I call you Joe for the purpose of this episode?
Yeah. I usually ask people to call me Joe anyways.
Cool. So Joe is a well-published, that's an understatement, and well-trained cancer biologist. His research focus up until now has primarily been to understand the metabolic, epigenetic, and therapeutic strategies for a number of cancers, including blood-based cancers like leukemias and breast, ovarian, liver, and more. Joe is a wet lab scientist, which means that he spends most of his time in a lab working with diverse cancer cell types and animal models to better understand and treat human cancers. Currently, Joe is working at a startup biotech company investigating the potential for targeted alpha particle radiotherapies to treat solid human cancers. And I'm just going to give you all a heads up that just right before we hit record, Sarah and Joe were just like riffing casually, and I understood like maybe 20% of what they were saying.
Don't worry, though.
So I'm going to be doing a lot of listening and learning.
We're going to bring you all into the wonderful world of cell biology today.
Absolutely.
So no, the only thing that I bring value here to this episode is I'm just going to set the stage with some statistics. And all jokes aside, there's been a lot on social media. Sarah, of course, as you all know, is not on social media, but Joe is very active on social media. And Joe, wait, what is your handle, just so folks know where to follow you?
It's Dr. Joe Zundell.
Okay, and we'll tag this in the show notes.
Yeah.
But the big question is, okay, we're seeing this increase in cancer diagnoses. What does this mean? What's driving that? And it's understandable. I'm sure everyone listening right now either knows someone, has a loved one in their family or a friend or someone who has had cancer. This is something that touches all of our lives. So just to set the stage with some statistics, we won't go too in depth here because there's a lot that we're going to learn from these two biologists right now. But okay, so half of all men and a third of all women in the U.S. will be diagnosed with some form of cancer in their lives. And around the world, out of every 6 people who die, one of them dies of cancer. Let's see some other stats. So cancer is among the leading causes of death worldwide. Okay, so in 2022, there were almost 20 million new cases and almost 10 million cancer-related deaths worldwide. By 2040, the number of new cancer cases per year is expected to rise to just under 30 million, and the number of cancer-related deaths to 15.3 million. Lots of differences, of course, by demographics. Cancer rates generally are highest in countries whose populations have the highest life expectancy, education level, and standard of living. But for some cancer types, such as cervical cancer, the reverse is true, and the incidence rate is highest in countries in which the population ranks low on these measures. Just a couple of other things here. The most common cancers listed in descending order, according to estimated new cancers in 2024, are breast cancer, prostate cancer, lung and bronchus cancer, colon and rectum cancer, melanoma of the skin, bladder cancer, kidney and renal, pelvis cancer, non-Hodgkin lymphoma, endometrial cancer, pancreatic cancer, leukemia, thyroid cancer, and liver cancer. I feel like there's a million stats here. We could put some more in the show notes. I'll stop there. Needless to say, there's a reason we're talking about this topic. So Sarah, you love a definition. Let's set the stage here before we turn things over to Joe. What is cancer?
All right. Well, cancer is a little bit different from when we're talking about other types of diseases like a viral or a bacterial infection, because with diseases like that, it's something attacking your immune system in your body. But when we're talking about cancer, what's tricky about cancer is that it's us, or it's like it's too much us. And basically, our bodies are a cell, like a factory of trillions of cells, right? And these cells normally grow and multiply to form new cells like as they're needed. And cells, over time, over a lifetime, can become damaged and they die off, and our body then replaces these cells in a process called cell division. And this is a really important process because our body can then replace the damaged cells with new ones, and this is part of what keeps us healthy and maintains homeostasis. But the thing about cancer is that cancer is when a human cell divides uncontrollably, and it attacks us because the cells can't stop doing this. And what happens is the cells will divide, and they'll cluster in these masses called tumors, which can grow in nearly any part of our body. And what's really important to note, and I think Jesse alluded this when you were talking about the different types of cancers, is that cancer isn't just one disease, but it's a class of diseases, a collection of different diseases. And it's caused by just a combination of many different genetic and environmental factors and the integration of genetics and the environments. And this is what kind of makes it so challenging to study. So I have all the respect in the world for cancer biologists like you, Joe, because I just study the brain and I study Alzheimer's disease, which is one disease, right? And so today I just wanted to pick your brain about a specific way of conceptualizing cancer biology that I think was first talked about in 2000 with a seminal paper. And what it is it's the hallmarks of cancer. So can you talk us through what those are and why it's important to discuss the hallmarks of cancer when we're talking about cancer biology?
Yeah, so as a cancer biologist, obviously our whole goal in our training is to gather enough evidence from a biological perspective to know all of the sorts of things that can happen in any given cancer type. Because obviously cancers are very diverse across the organs that they originate in. And so over time, from a very long period of time of just collecting data, no matter how primitive it has been, we've come to understand there's a lot of commonality between cancer types. And in terms of mechanisms, these are called the hallmarks of cancers. So in about 2000, the year 2000, Douglas Hanahan and Robert Weinberg, which I actually have Robert Weinberg's shameless plug cancer biology book. It's the best cancer biology book for those cancer bio nerds out there. Actually, shout out to Justin Bonfini who gave me this book as a gift. It's actually a little bit pricey, but Douglas Hanahan and Robert Weinberg came up with some of all of the known mechanisms, which they've called the hallmarks of cancers, that are known across all different cancer types. So in this case, there's 14 of them that we've gathered over the years. There's actually 3 hallmarks of cancer papers. The first paper contains, I think, 6 or 7. The second paper adds 3 or 4, and then the third paper adds a couple more based on things that we've learned. So I'm just going to list them off real quick. Of the 14, there's sustaining proliferative signaling, so things that maintain continuous cell growth, which is kind of what Sarah had alluded to, which is a characteristic of cancer. Evading growth suppression, so there's things that tell a cell to stop growing, as there are things that tell cells to continue growing. Cancer cells resist those urges to grow and stop growing. There's also resisting cell death is a hallmark of cancer. Obviously, cell death is a very important part of life. Sarah mentioned homeostasis, or a state of balance, earlier. And we need to maintain a certain number of cells and an appropriate integrity of our cells to be healthy as we age in life. Another one is becoming immortal, or enabling replicative immortality. cancer cells will mess with cell division processes and do some weird things to chromosomes, which can cause problems, but those actually enable them to continue growing. We can talk more about that later. There's also angiogenesis, which is a normal process in our body, but cancer cells kind of hijack blood vessels. And they don't do it in such a great way. It's not superior, but basically cancer cells will recruit blood vessels nearby to grow towards the tumor to supply it with oxygen, which is usually necessary for cancer cell growth. But it comes with problems. And then another hallmark is invasion and metastasis, which is typically seen in later stage diseases. Cancer cells essentially need to pick up and pack their bags and move to distant organs, and they need to adopt and change certain characteristics to be able to pick up their bags and move to a different organ and then plant and survive there.
And Joe, sorry not to interrupt you, but those were the 6 that were in the original Hallmarks of Cancer paper, correct?
Yes.
Okay,
Yes.
so maybe we can start off by just taking those 6 and going in maybe into a little bit of detail one by one.
Sure.
And then we can mention the rest of them
Yeah.
as we go on.
Part 2.
Yeah.
Part 2,
Cancer
or maybe
daddy.
Part 4.
just so we don't like overwhelm people with the information. Like let's break this down a little bit.
Sure.
And wait,
yes.
can I just call out that what you both are doing right now is actually really impressive, because obviously these are very complex topics, and so they're going to try to make this understandable for someone like me. Although I will say I did minor in biology. I don't know if you know that, Sarah.
I did.
But yeah, but let's just say a lot of this is over my head. So yeah, so Joe, if we can go through some of these, we'll see how far we get. That would be awesome.
Absolutely. And as I told you guys beforehand, and as we've discussed even offline, it's super important that we try and break things down for as many people as possible to understand, because it's also important to note that sciences are very diverse. There's going to be people, even within cancer biology, that don't understand the type of cancers that I study.
Mm-hmm.
So the simpler that I can explain those things to even people in my own field, the better equipped they will be to address tackling other problems and other types of cancers.
Yeah, and I think
So
an
it's true
important
for
thing
people
to
outside.
note when we're talking about these types of mechanistic hallmarks of diseases, I mean, cancer is such a heterogeneous collection of diseases. So, I mean, this is really just a framework of things that just like broad things that like all cancers really have in common. Right, Joe?
Yeah, exactly. And that's why they're called the hallmarks of cancer. So first starting with sustaining proliferative signaling. It's just big fancy words for cancer cells keep growth signals on, because that's what a cancer is. A cancer cell wouldn't be a cancer cell if it wouldn't continue growing. So oftentimes the way that cancer cells will sustain their growth is by increasing the abundance or expression of a variety of different proteins on the surface of the cells, or even metabolic enzymes within the cells, and keep those signals on. Because obviously if those signals are off, and this is a dramatic oversimplification, if the signals are off, then obviously it can't continue growing. And it's also important to note that these signals are very, very diverse across other cancers. So despite one cancer like breast cancer having, the HER2 receptor, many people have heard of the HER receptors or progesterone receptors being highly expressed on the cell surfaces, maintaining growth signaling in breast cancers. Another cancer like lung cancer might increase the expression of an epidermal growth factor receptor, as an example. So there's many different growth pathways across cancers that change their growth, kinetics of those cancer types, and of course changes. Somebody doesn't like cancer.
I'm so sorry. My dog's barked. Yeah. Sorry,
That's okay.
guys. Sorry,
anyways,
Joe. Yeah.
No, it's okay. So just keeping that simple, every cancer has its different growth pathways, and of course, because of that, requires different treatments. Does that make sense?
It does make sense. I have a question, but I don't know if I should ask it
Ask
or if
it.
I'm going to take us off track here. But so you're talking about, and I know this is an oversimplification, but this being turned on. So what turns that on? What are the factors? And then are there modifiable things, non-modifiable things? Because obviously everyone wants to know, how do I prevent cancer? Could you just talk a little bit about what turns things on or off?
No, it's a good
So
question.
Yeah, no, that's an awesome question. And there's a million answers for it. There's a lot of genetic contributions which can enable genes to be expressed, largely driven by environmental considerations. Now, when I say environment, what I mean is also nutrient adaptation. So the available oxygen in an environment will change gene expression. The available glucose, amino acids, basically the macromolecules of life will signal the genetic framework to enable basically DNA to be read and enable genes to be expressed to make some of these proteins and enzymes that respond to those things to enable a cell to grow. And now these are always in normal cells too, but what happens in cancer cells is sometimes there are mutations that occur that can, for example, there's a common mutation amongst a variety of different cancers called PI3 kinase. And sometimes these mutations of this enzyme can enable independent of the level of expression. So you don't change the abundance, but what you can do with this enzyme is you can mutate it and change the activity. So basically the enzyme is hyperactive. And so that's what keeps these growth signals on from a mutational perspective that's developed in cancers that have this specific mutation of PI3 kinase. And that was actually a subset of my thesis work was trying to elucidate targeted strategies to counteract that mutation in ovarian clear cell carcinoma.
Very cool.
But
So
again,
if I could just rephrase
Sure.
to try and maybe summarize a little bit here. So sustaining proliferative signaling in a normally dividing cell, right? There's a signal that tells the cell like, yep, go ahead and divide, like thumbs up, we're going for it. The cell
Yep.
divides, and then there's another signal that says like, all right, time to stop, like we've done the thing, like we're good now. Enough
Exactly.
is enough. But with cancer, and what this first hallmark is laying out for us is that when it comes to sustaining proliferative signaling, is that like off switch, that like little like signal that says like, all right, cell, like we're done dividing, like we've done what we came here to do, like, and now stop now. It just doesn't, that signal doesn't happen, right?
Exactly. So it's basically cancer cells become ignorant of the rules that govern life. Because obviously, again, wouldn't be cancer if they followed the rules of life.
Yeah. I've heard
And that actually
the
leads,
like analogy of like kind of disobeying us, like a stoplight.
yeah, only inside of our cells we have hundreds and hundreds of different mechanisms as checks and balances. So oftentimes people, I think, have this assumption that life is a very simple binary box of a few things that control a lot of things, but life is very intelligent. Our cells are very smart, and we've evolved a number of different ways to counteract errors if something fails. So when it comes to DNA repair, we have things that respond to double-stranded DNA damage versus single-stranded DNA damage. When it comes to enzymes, we have feedback regulation. So if an enzyme is making, for example, in glycolysis, when you take in sugar from your diet, as an example, it's brought into cells. There are feedback mechanisms as glucose is broken down to signal back to enzymes in the pathway that the glucose has reached to a certain point in the degradation or breakdown of glucose for energy to tell some of the enzymes to stop. So there are rate limiting steps that help to control that in enzyme cascades. And cancer cells find creative ways to ignore those things. And it's not a perfect system. And that's what creates errors, and that's what eventually enables cancer patients to become very sick because their cancer cells essentially are ignoring these rules of biology from a variety of different perspectives, and they're essentially becoming very selfish, even amongst themselves.
Yeah.
Does that make sense?
Shockingly, it does make sense. Thank you.
Good. That means we're on track.
Yes. Okay. So now, are we still on the first fundamental?
That actually segues quite nicely into the evading growth suppression.
Yeah, I was
Okay.
going to say 1 and 2 really go hand in hand, don't they? They're kind of two sides of the same coin, as I understand it, right? Like,
basically, yeah. So the same things that promote growth are often involved within suppressing growth. And this is where the feedback loops come into play that I was just talking about. So a lot of proteins can have double-edged functions in maintaining growth and growth suppression. When it comes to growth suppression, a good thing to talk about here is P53, because P53 is called the guardian of the genome for very good reason. It's
I
a
love
very
that.
important, yeah, it's a very important protein that responds to DNA damage and repair. And so again, in normal cells, P53 is responding to errors that happen naturally over time. Some of these errors even stimulate evolution. But if P53 can't repair those things, then that's when you increase the likelihood for mutations that sometimes can be favorable and sometimes maybe not favorable. And in some of these cases, they can relate to the development of precancerous cells. And that's why cancer cells, P53, it's known as a tumor suppressor, because if cancer cells can shut off or reduce the function of P53 from repairing DNA, cancer cells can grow. So it's shutting off a tumor suppressor.
I'm jumping in here with a real life story that I think illustrates what you're describing right now.
Perfect.
Probably TMI, but that's my jam.
I love it.
Pap smears. So I have had pap smears that have come back. I had HPV at one point. I know I've shared that on the pod. They did a colposcopy or whatever they're doing. They're testing the cells in there, and it came back that I had pre-cancer cells. Obviously, this scared the poop out of me. Then I kept getting, more pap smears, and then eventually they're like, "Oh, you're all clear now." So does that? And the same thing, by the way, happened with a lump I found in my breast. I am Ashkenazi'd out over here, but yeah.
Same.
So yay! Oh, we got a little. Oh, sorry. We had a little triple Ashkenaz situation
we got
here.
The Jew Club going on here.
Yep. J
Cool.
Club. All right, sorry. But so is that sort of just for folks who are listening who maybe have heard similar things like, oh, you have pre-cancer cells, but then you're cleared. Is that sort of what you're describing here? Like the body is repairing?
Yeah, maybe we should also just define in general what is pre-cancer.
Yeah, pre-cancer is a state where it's not cancer. It's pre-cancer because there are multiple events that need to happen in most cases, not all cases, for a cancer to initiate. And so when something is designated as precancer, it's because we commonly see that thing in a variety of different cancers being mutated. So if we see P53 function is let's say P53 function or some other enzyme is mutated in cells, or there's some weird trend that we're seeing, it can be indicative of those cells being messed up in a way that could lead to cancer, but it's not always the case. So something I always like to talk about, and this relates to this growth suppression thing, is the two-hit hypothesis. And again, it's not always the case, but it's this hypothesis that has stood pretty well in cancer biology that would require more than one event to initiate cancer formation. So precancerous event would be like the mutation of an enzyme, something like KRAS or P53, which is a DNA repair enzyme or protein. And sometimes those mutation events or changes in expression can cause a snowball of events that initiate other things to happen, or you can interact with a carcinogen or some sort of environmental stressor that attacks a different angle of cell biology, let's say radiation or something like that, that basically creates another hit, which now can create more snowball effects and less regulation of cell division and control. And that's typically when cancer forms upon the two-hit hypothesis. But even then,
and I imagine it's probably often more than just two hits.
Exactly. Yeah. And in some cases, we're finding that a single spontaneous event can be sufficient to cause other events. So while it's not quite the two-hit hypothesis, depending on how severe the first event is, it can cause a chain reaction of things that can initiate tumor formation.
Sure. Yeah, like a biological cascade.
Exactly. And everything in biology is a cascade anyways.
Right.
But
it's a good way to think about
again,
things.
This inability to follow these checkpoints and these rules that when not repaired by these checkpoints and repair systems can initiate tumor development.
Yeah. So just one more crack at like just summarizing the concept of P53, which you emphasized was like very, very important. In context. Sorry, Jess's dog.
That's okay. My
Um,
cats will probably show up at some point.
I love this dog so much. We really bonded when I went to visit Jess a couple weeks
He's
ago. All right. Anyway, sorry. Okay, so P53. P53.
Mhm.
This is called a tumor suppressor gene.
Yeah.
And basically what that is it just why it's so important is it tells cells to stop dividing, right?
Mhm.
So it prevents the
In certain
creation of
circumstances.
tumors, which happens when cells divide uncontrollably and clump together. That's a
Yeah.
tumor, and P53 stops that from happening, right?
Yes.
So-
And it can do that, which leads us into the next hallmark of cancer by initiating cell death. So P53, as the guardian of the genome, if it's detecting issues within DNA repair mechanisms, it will signal a process called apoptosis by forming complexes with different proteins. And those proteins with P53 will bind to different regions of DNA and signal specific proteins that tell the cell to undergo what I said is apoptosis, which is a programmed way of the cells dying. So it's, I guess the simplest way to say this is like very controlled suicide. So it's not like an immediate cell death, because in biology, immediate cell death or necrosis is a very, very bad thing to happen. So it's a very organized form of cells dying. And it's really it's very interesting to watch under a microscope. Cells will literally form like these little bubbles or blebs off of each other, and organelles will, be broken down and recycled.
Yeah,
It's really,
I
really
mean,
interesting to watch.
what's so cool about apoptosis specifically is that like it's like you said, it's a very controlled process. So
Yeah.
it's not like a wasted death. It's that other cells in the vicinity can use some of the sort of debris from that dying cell then
Exactly.
to help themselves.
Mm-hmm. Exactly. And so, in cancers, when you have like a mutation in P53 as an example, and there are very different types of P53 mutations. If the cells can't, how do I say this? If P53 function is messed up in cancer cells and they can't undergo cell death, they will continue to grow. If P53 can't signal to the cell death machinery, hey, there's something wrong with the DNA here, I can't repair it, stop growing, cancer cells will mess P53 up and they'll continue growing and resisting cell death. And another way that they do this too is by increasing the expression of proteins within the cell death machinery that are anti-apoptotic. So there's anti-cell death stuff in cells, and there's pro-cell death stuff in cells. Again, these are checks and balances,
Which is
even in
important,
cell
right?
death.
Because like we don't want our healthy cells to be dying and undergoing
Exactly.
programmed cell death. So like it's important that we don't have like the apoptotic signals going crazy in all of our cells at all times.
Exactly. So oftentimes in a lot of different types of cancers, they'll increase the expression of proteins like BCL2 is a protein in increased expression in many different cancer types, let alone ovarian cancer, which is what I had studied quite well in my thesis. And so we've actually developed inhibitors against this anti-apoptotic protein because cancer cells will increase this pro-life molecule, which prevents the cells from dying. And if we make an inhibitor against that, which we have, the FDA-approved venetoclax, which binds to BCL2 and prevents it from keeping the cells alive, and it basically topples the balance in favor of cell death versus pro-life. So inhibiting that protein in cancers has been pretty effective.
I have probably a stupid question. Can I ask
It's
a
not.
stupid?
So first of
No,
all, never say that anything you have is a stupid question.
you know what? You're right.
Yeah.
You're absolutely right.
But
I
don't
take
do
it
it again.
back.
I will scream at you.
won't ever do it again.
This
Okay.
is so complicated. This is cancer biology. Like this is much, like I said earlier, much respect for people that study this because oh my God, it's complicated.
So we want the bad cells to go through this apoptosis, right? Okay, yes.
Yes.
We don't want the good cells, at least we don't want them to prematurely go through apoptosis. Am I saying that right?
Correct.
Apoptosis?
Yeah.
What is that if they do? If the good cells die prematurely, is that cancer or is that something different, or am I making something up?
That might be more relatable to like an autoimmune disease.
That's what I was
Okay.
thinking.
There are
Okay.
and I'm not an expert in this. Dr. Yasmin Mohseni might be better
Love
equipped
her.
to discuss that. She's amazing.
She's amazing. Yeah.
And there's a variety of other immunologists on social media that can discuss that. I don't know how accurate that is to relate it to autoimmunity. I have
Okay.
taken a lot of immunology courses to study this stuff, but it's not going to be as dedicated as some other people who are much more qualified than I am in that realm.
But really, so what I'm hearing is, so really we're talking about the bad cells not dying when they're supposed to be dying and sort of overgrowing or accumulating. And that's what we're talking about with cancer. Okay.
Yes.
Yeah.
And your body can
Scene.
clear that up to a certain point too.
Okay.
Because obviously there's a difference between benign tumors and malignant tumors. Yeah.
When
but
do we get to malignant? But I don't know if that's coming up in the discussion.
That's always really challenging too, because it's also something that confuses cancer biologists. Because the things that make benign tumors, they're still actively dividing, but they're not doing it in such a way that is super damaging to your body.
Well, does this have to do with immortality, the establishment of immortality in cells?
Yes and no. So nice segue, Sarah. The next hallmark of cancer is
See what you did there.
enabling replicative or immortality. And so what Sarah was referring to there, somewhat vaguely, to transition us to get caught into a loophole of talking about the same over and over. Excuse my language.
Bleep.
Yeah. So there's a certain amount of times realistically that a cell can divide in a healthy way. And actually, this is something that's called the Hayflick limit, which was described by Leonard Hayflick, actually, where I did my thesis work over in the UPenn area at the Wistar Institute. And so Hayflick had described in his experiments that there's a certain amount of times that a cell can divide well to where it eventually becomes senescent, which is senescence is also another hallmark of cancer, but it stops dividing basically or undergoes cell death. But cancer cells will surpass this limit because they've acquired so many issues within cell growth, or preventing growth suppression, or resisting cell death, that they just continue to grow. So when it comes to understanding replicative immortality, it's more of maintaining chromosomal generation over time.
Mm-hmm.
So within our cells, we have DNA, and DNA is organized and bound up by proteins and coiled together into chromatin via histones into chromosomes. And chromosomes, as our cells divide, need to be very, very carefully copied via mitosis. And there are enzymes that preserve the integrity of chromosomes. One particular enzyme, super important, is called telomerase. So at the ends of chromosomes, there are these DNA repeats. And telomerase's job, this enzyme, is very important. It's to preserve the integrity of the ends of the chromosomes. Because as we age, as cells divide, those repeats become smaller and smaller. And when they become so small, it signals the cell to undergo cell death. Because there's only so much genetic material that can continue to propagate the life of that cell. But cancer cells will increase the expression of telomerase to a point where they will maintain chromosomal reproduction between cell division, even if there are errors present. And this can, but not always, lead to another hallmark of cancer, which is genomic instability, which we'll actually discuss next time. But it's essentially the accruing of genetic errors that are often related to replicative immortality. because cells are only supposed to divide a certain number of times.
Right. And it's also important to note here that the number of times that a cell divides typically depends on the type of cell we're talking about too. So like certain cells will divide naturally more are more inclined to divide than other types of cells.
Yeah. Do you want a good example of that?
Yeah.
Hair cells have a high turnover rate. Obviously, we're actively growing hair as mammals. And that's one of the reasons why, as an example, we see hair loss in cancer patients from chemotherapeutics, because the drugs that we develop are targeting these growth pathways in cancer cells. And because hair follicles have a high turnover rate that sometimes match the growth rate of cancer cells and use similar mechanisms as to what the drug is using, that's why hair follicles are targeted by some of the chemotherapeutics as well.
Wild.
Mind is blown.
Yeah, so there's a lot of overlap, and it's hard to, make drugs super specific to a process without impacting healthy cell processes.
Yeah, I mean, I imagine that's why chemotherapy is often likened to, poison.
Depending on the drug. And I kind of hate that it is likened to poison, although I'm empathetic and I understand why. Because, after witnessing firsthand, what cancer can do to somebody, my mom passed away from esophageal cancer, for those that don't know, when I was 17 years old, and seeing, what these medications can do to humans, let alone those who have cancer, is shocking. But it's always a double-edged sword, because nothing in biology is an either-or situation. And so no matter how hard we try to make these therapies targeted, there's always going to be some overlap in biology, because as Sarah pointed out in the beginning of the episode, cancer cells are us to some extent, right? Until they aren't, until they've evolved in a way that they're so messed up that they're competing with us. It's not an integral part of it's not favorable to the evolution of our bodies or health in general. So again, while we try and make specific targeted therapies, there's always overlap in biology, and therapies have gotten a lot better over time to become even more targeted, but it'll never be perfect. There's always going to be trade-offs, and that's also why we try and combine therapies together to try and reduce the likelihood that these things are going to affect healthy cells more than they are cancer cells. Does that make sense?
Yes, that was beautifully put.
Okay.
And so just in the interest of time, I think maybe we can cover a few more of these hallmarks, maybe the next two more, and then that'll sort of close the book on the original 6 hallmarks that were established. And so talk to us, Joe, a little bit about angiogenesis. What is that?
Yeah. So actually the next 2 hallmarks, which are angiogenesis and invasion metastasis, kind of play into each other a little bit, so they segue into each other nicely. Angiogenesis is a normal process in our body. Angio meaning blood vessel, genesis meaning, the production of new things, new blood vessels. It's a normal process, again, very tightly controlled, because you don't want blood vessels just growing anywhere in your body, because you look like a weird bloody tree. Which we also kind of do in general, but like a really gross bloody tree, which is actually what a tumor looks like in many cases. But angiogenesis is a hallmark of cancer, because cancer cells will secrete signals to the environment because they're competing with their own resources. They need resources from our bodies. So they secrete signals. A lot of them are specific proteins that tell nearby blood vessels to grow towards the tumor. And that is in efforts to supply things like oxygen, amino acids, proteins, sugars within our bloodstream to the cancer cell directly, because a lot of the time in developing tumors, those nutrients are beneficial for their growth. But the trade-off here is that in creating those blood vessels towards tumors compared to normal healthy blood vessels, cancer cells make really crappy versions of a superior product, which our body normally regulates. So actually, one of the really interesting things when I was learning cancer biology in focus studies in grad school, a professor showed us the difference between normal blood cells or normal blood vessels and those that grow within a tumor. And the ones that grow in a tumor and around a tumor, because the signals are dysregulated that grow those blood vessels, they're leaky. They have poor, to maintain blood pressure. We need certain wall integrity of blood vessels. They're very poor integrity regulating pressure amongst the blood vessels, so they collapse inward often, especially as they grow into solid tumors, where tumors, people may not realize this as well, they're more than just one type of cancer cell. It might be one solid tumor, but depending on where a cell is within a tumor, it will have, it's essentially a different cancer cell. It adapts to the nutrients within its area. So there are oxygen-rich areas and oxygen-depleted areas. And this is what's called hypoxia, which is low oxygen. And so that's why these low oxygen cancer cells are sending signals to the neighboring environment to bring blood vessels towards them so they can get more oxygen, because oxygen is incredibly important for maintaining the metabolism in like the mitochondria, as an example, for what's called oxidative phosphorylation, the electron transport chain. A bunch of jargon that we don't really need to get into.
Yeah, we don't need to. Let's not touch the electron transport chain right now. Like,
yeah. Can you help me understand something? I'm sorry,
sure,
Joe. I don't want to.
No, go for it.
So you're talking about these. I'm picturing these like trees. You said bloody trees. They're vessels that are bringing the blood, right? Or is it the? Yeah. Okay. And when we're dealing with cancer cells, they're not great, right? You said they're crappy, they're not structurally great. So I'm just thinking through, like, what is the clinical implication of that? Or how is that like feeding the cancer? Is it what you were just saying, that because they're transporting things poorly, you were just saying something at the end there where it's almost like, is that leading to, and I'm probably saying this, butchering this, but like feeding the cancer cells more? Because is that sort of what you were getting at?
Yeah, so essentially and I've said this because everything is a trade-off, and in cancer, it's more about sacrificing the efficiency of a process for speed. And so that's why they make these poor quality blood vessels. And from a clinical perspective, because they're making these poor blood vessels very quickly, they're often relatively easy to target. So we've developed therapies to go against these angiogenesis signals. So one such therapy was actually used to treat COVID, believe it or not, because of the vasculature in the lungs being affected, which promotes inflammation. There's this protein that is secreted by cancer cells called vascular endothelial growth factor, or VEGF. And so we have this, it's an antibody that goes against VEGF. It's a humanized antibody. It's called bevacizumab, and it's FDA approved to basically grab VEGF and prevent it from stimulating angiogenesis. And so when the antibody binds VEGF, it will get sequestered in or build up in your blood and get excreted, as opposed to promoting, it'll block it from getting to the cancer cell.
Here's my question though, but don't some of our cells need VEGF then? So how does a therapeutic like this prevent, like selectively target the angiogenesis that's occurring within the tumor, as opposed to that which is occurring, like, for example, in the blood-brain barrier?
Absolutely. And that's a super good question.
Probably a big one too.
No, there's a couple of physiological considerations regarding antibody size and getting into the blood-brain barrier relative to VEGF size. Antibodies are quite a bit larger, I think, than VEGF. Don't quote me on that, but I believe they're quite a bit larger, and I think they're challenging.
Yeah, they're definitely larger.
Yeah, it's challenging across the blood-brain barrier. The other thing is, it comes with a trade-off, right? So we don't want to give this therapy for a super long period of time, and we have to know that cancer cells are secreting VEGF at a rate that where we give bevacizumab, it's going to impact the cancer cells more than it is healthy cells for a specific period of time.
Right. So what I'm hearing is that like balance is very important when it comes to these types of therapeutics, and like that's
Absolutely.
why.
And there are negative health effects, again, for any type of therapy. no matter how targeted we get. And this is especially true in the immunotherapy space, as we've learned. No matter how targeted we make against a therapy against a very specific molecule on a cancer cell, from an immunotherapy perspective, your immune cells also need to control the way that they're reacting against things. Too much of something can be a bad thing. So if I have my immune cell acting against a cancer, from an immunotherapy perspective, it'll do a really good job, but if it does too much of that job, it can cause other problems.
Because
And
cancer
we won't
cells
get into
are
that.
still our cells. They're still us.
Right. They are up until a certain point. And that's one of the other hallmarks of cancer, which is unlocking phenotypic plasticity, which essentially cancer cells are not you anymore.
Oh.
They're you, but they're so messed up that everything has a path in life. And because of those level of organizations in our biology, sometimes those things are very strict. But when something like a cancer cell becomes so messed up, it can go backwards on a path or go on to a different path that it's not supposed to be on.
So maybe this is a good time to talk about like invasion and metastasis.
Yeah, because that goes down a different a rabbit hole, but it's also kind of similar to what needs to happen for invasion and metastasis. So we had just spoken about angiogenesis as an example. Cancer cells will secrete signals to enable blood vessels to grow towards tumors, but cancer cells will also adopt characteristics as they're enabling blood vessels to grow towards the tumor to, what I said at the beginning, pick up their stuff, go into the blood vessels, and go to distant organs, unpack their stuff, and make a new home. And that's very oversimplified, but the processes that govern cancer cells' abilities to metastasize or get away from their primary tumor, invade other tissues and go to other tissues and actually like sit there are many. And there's a lot of mechanistic ways that they can do this. I can get into them briefly, but this is why metastasis is something that's seen in later stages of diseases, because there's many characteristics that a cancer cell needs to change about themselves, such that before they can even pack up, they have to be able to kind of like think or be sentient enough to know that they'll survive somewhere else. And that's why we see specific cancers, even though they've metastasized, they'll only go to certain organs because they only have the appropriate framework to survive in specific organs. Does that make sense?
Yeah, that does make sense. I mean, it makes sense that it would occur in later stages too, because enough of the cells in a given location would need to amass, right, in order for them to just be like, all right, yeah, I'm gonna like take a little vacation. I mean, I'm gonna go to a different organ system, or like a different part of the body, because like the tumor must have had to progress enough for that to happen.
Yeah,
just from a sheer volume standpoint of the number of cells.
Yeah, exactly. And it's really important for people to realize that normal cells move around all the time, but they won't get up and go to different organs.
Right.
And that's why it's it's a programming that needs to happen for metastasis to happen. So very, very simply, cancer cells will increase enzymes which break down these sticky proteins at the surface of the cells that kind of keep them in place like a foundation of a house. They'll increase these production of these enzymes that break down the foundation of the house such that they're not sitting on other cells anymore, or even collagen matrix within cells, within tumors. And then they will, once detached, they'll respond to the fact that they're now floating around in space. They'll respond to that by increasing the expression of a number of different proteins to make them more malleable, the cells more malleable. Because when they were sitting down on something, they were a little bit more rigid. Now, once they're up floating in space, they need to be more malleable. And that's especially true if they want to get into blood vessels, because they have to push through it. And this is something called extravasation or intravasation. So they have to squeeze through these tiny little holes to get into blood vessels and travel to distant organs. And then once they get to those distant organs, they then need to reprogram themselves such that they can increase the surface expression of these sticky things to then stick down to a new surface, and then change their metabolism to adapt to the new organ.
I have a logistical question.
I'm
So,
sure you do.
well, I mean, a million questions, but
Yeah.
actually, logistical is maybe not the best word. I'm so curious because you're saying metastasis, which obviously, like I, as a person who's not a cancer biologist, like I'm thinking we're getting towards the end of the discussion there. Like that's what everyone's scared of, cancer metastasizing, right? Just in terms of these fundamentals, is there logic to the order in which we're going? Because again, like I would think this would be toward the end of the list, just with my lay understanding of cancer. I'm just so curious about that.
Yeah, so I would probably, that's a super good question. I would probably place invasion and metastasis and unlocking phenotypic plasticity at the end of the list because they are late stage
Okay.
events.
Mm-hmm.
Because to me, they're kind of similar, but also very different in a way from a mechanistic perspective, which is why they're separated.
Got it.
And you know, not to scare anybody, but typically when a cancer does metastasize, the treatment options, because the cancer cells become so messed up, the treatment options are abysmal. Like
Right.
I don't have any other way to say that.
Which is
Um,
why it's my understanding that there's a push in on the clinical side of things for early detection when it comes to cancer, because when you're in those earlier stages prior to invasion and metastasis, it's a little bit easier to organize some of these therapeutics in a way that's actually effective.
yeah, exactly, and that's one of the reasons that prompted me wanting very much to get into my current job. So earlier in my introduction, you'd mentioned that, I'm working for a radiopharmaceutical company using alpha particle therapies. But another portion of my job currently is using these radioactive isotopes as a diagnostic tool as well. Um, so a lot of people have heard of PET imaging and those sorts of things. Basically, what we can do nowadays is we can make a molecule against a specific biomarker in a cancer cell. So let's say, you know, lung cancer cells increase EGF expression. I can make a molecule against EGF and I can strap a specific radioactive isotope to it. And I can use PET to see where those tumors expressing EGF are. And they will light up brighter than normal cells in a PET scan or maybe even something like SPECT CT. There's a bunch of different imaging methods. Um, but nowadays we're getting a lot better at using these targeted radioisotopes to better diagnose cancers even in earlier stages.
Mm-hmm.
So the earlier you can diagnose a cancer, your chances of treatment for that cancer, despite even any negative outcomes due to the therapy or even having the cancer, are going to be so much higher than when cancer cells have the time to develop the characteristics to metastasize. Because once it does metastasize, they're kind of too far gone.
And this is, I mean, now you're really speaking to my public health heart. I feel like this is now the intersection of our worlds, right? And why folks like me, we really emphasize the early screening, and why many of us say perhaps it doesn't entirely explain the trends that we're seeing in increased
Sure.
diagnoses, but increased cancer rates, but it likely is a result of the fact that we are pushing early screening and things like that. It's detection bias. So anyhow, I know we covered a lot of ground here, and I know we have a lot more ground to cover. Any last thoughts just to wrap up the discussion of these first 6 fundamentals of cancer, Joe?
Yeah, I mean, my last thought is for people to understand that while this stuff is very complicated and it's very easy to place these things in kind of a binary box, and say, oh, we can just target angiogenesis, we can just target replicative immortality, we can just target cell death. Cancer cells will use all of these hallmarks at some point in their development. And of course, if we can get things before they metastasize, the better outcomes of treatment will be. But I think it's super important for people to realize that as a disease, it's really challenging to treat in a way that you can get the cancer cells more than you can healthy cells. And we have to be very, very careful about, where we get information online, because there are just a lot of people that make this so simple to the point where it's actually damaging to a person's health. I mean, we've all heard people say, cut out sugar, eat just, ketogenic diets. And, there are contexts where these things can be helpful, but we don't understand enough about them right now. And giving advice like that to people online, especially cancer patients, without understanding what they're going through, without understanding their treatments, without understanding their current quality of life, without understanding whether or not they can engage in physical activity to exercise, what their sleep health is like. If we don't understand these things, we're giving blanket advice that is way more likely to do more harm than it is to do any good. And so without access to a person's medical health records, which I can't legally acquire online anyways, and I shouldn't be, I can't realistically make appropriate clinical decisions for a lot of even the patients that come to me. And I tell them honestly, like, I want to help you. I want to give you good outcomes. I can give you suggestions based on information that you give me, but you have to go back to your oncologist and say, is this a realistic consideration based on my discussion with this cancer biologist? And they will tell you, based on your medical history, whether or not that's feasible. And if it is, I've done my job. But you have to work with your medical healthcare team to enable these decisions. You can't just trust, Thomas Seyfried online because he said, oh, fasting is going to cure cancer. I have all the secrets. The NCI is lying to you. And yet I'm a professor at Boston College and understand biochemistry somehow. Or Walter Longo because I posted a paper on fasting and I charismatically talk about that online too. Or
Joe's naming names.
I know,
Yeah,
let's
naming names.
there's a bunch of people that do this. And again, while they do good work, sometimes this level of charisma is damaging to people like me who are trying to give people a reasonable framework to understand this disease, because it's not as simple as they make it. I wish it was, because if it was so simple, I'd be a trillionaire. I would have all the answers to treating cancer.
Right.
But because it's so complicated and there's a lot of overlap, I don't.
And I think that's the case in a lot of scientific disciplines and with a lot of diseases like that. It's frustrating for folks like us to see the oversimplification circulating online and the misinformation, which is why we're so thrilled that you were able to come on today and help us to really understand the biology of what we're talking about when we're talking about cancer, like at a really fundamental level. I think it was really important to have this discussion. So thank you.
Yeah, likewise, and thanks again for having me. And I will say this as a disclaimer, like I know there are many things I said that weren't entirely perfect discussing these mechanisms and these biological considerations, because when you try and simplify this stuff, it's very challenging to say things in a way that's going to be coherent. So that's why I try and maintain a high level of presence to answer people's questions, such that if I do say certain things that are confusing to people, I'm there to clarify. So people need to understand that I'm not perfect, but I'm here to help.
Love that. Well, you wear two hats. I mean, you wear this cancer biologist hat, but then you also do science communication. And it is so tricky, I mean, with a topic this complex, to capture the nuance, but still, make it understandable for folks. So thank you again, Cancer Daddy, Dr. Joe Zundell, for joining us.
Yep.
And I guess you have to come back for a part 2 because we've got more fundamentals to get through.
Should we just list them out right now?
Oh, the other things that are, do we want to just do it real quick? And then
we've got deregulating cellular metabolism and energetics, Thomas Seyfried's favorite target. Genomic instability, non-mutational epigenetic reprogramming, which is actually what my thesis work was on. Polymorphic microbiomes, because we're covered in bacteria, fungi, viruses, protists. We're disgusting. Tumor promoting inflammation, senescence, and unlocking phenotypic plasticity, and also avoiding immune destruction. So there's a lot of
Tune in
cool
next
stuff to talk
time.
about.
Buckle up, folks.
Tune in 10 years from now.
Oh
and
my
we
gosh.
still won't have all the answers.
Okay, well, on that depressing note, Sarah, I guess I'm going to start calling you Brain Mommy. Would you?
Do not.
Brain mommy.
All right. Would you like to take us home here?
No.
No.
Plaque princess.
Is that Oh, I think
Maybe
worse.
that's gross. That
I think
sounds
that
gross.
was worse.
This is horrible.
Okay.
I think we
That's
need to
worse.
shut this down right now.
Shut it down. Shut it down. All right, Sarah, take us home.
Thank you all for tuning in. We hope you learned a thing or two. I certainly did. I had such a blast in this episode. Please join us next week on your trusted source for no nonsense, just science.